Hello dear students, in this lecture we will discuss about the examples of rhizospheric microorganisms, and also we have discussed about the effect of rhizospheric soil on the microbes. So here large number of bacteria, fungi and actinomycetes are found in rhizosphere. Normally we can say 1 gram of soil contain million numbers of microorganisms. So it is nothing but a like that. Firstly, the major dominant microorganisms which are present in the soil, firstly bacteria. Many nitrogen fixing bacteria and phosphate solubilizing bacteria and other bacteria are found in the rhizosphere. So normally the beneficial association between plant roots and microbes. Normally we discuss about the nitrogen fixing, either it is symbiotic nitrogen fixer or non-symbiotic nitrogen fixer. Also phosphate solubilizing bacteria, as nitrogen and phosphorus are required for the growth and proper yield of that plant. So either they are available in the soil, but they are abundantly made available for the growth of plants by means of or by the help of that microorganisms. And environmental nitrogen is fixed by that nitrogen fixing bacteria and some phosphate solubilizing bacteria which make available phosphate for the growth of that plants. For example, Pseudomonas, Arthropacter, Azotobacter, Agrobacterium, Flavobacterium, Cellulomonas, Rhizobium, Clostridium. These are nothing but some dominant species of bacteria, or we can say some dominant genus of bacteria. Sometimes with respect to the when we collected different soil samples, species of that bacterial genus can be changed. So these are nothing but some representative sample of that bacteria. They are normally present in the soil and they have a beneficial characteristics for the crop. Another microbe, that is nothing but fungi. Some fungi are found associated with the root forming mycorrhiza and other occurs freely in the soil. Normally fungus can easily available, or we can culture the fungus from soil in or laboratory media, and they are normally present in the soil. Example of some fungus which are normally present in soil are also in association with the plant roots. We can say that is mycorrhiza. In this, ectomycorrhiza or endomycorrhiza we will discuss in next lecture. Some representative examples of this fungus are Marciella, Cephalosporin, Trichoderma, Penicillium, Glioidium, Glavomastric and Fusarium. These are nothing but the few representative name of genus of that fungus which are normally present in the soil. Either some are in association with plant root or for other purpose or for their life cycle they are carried out their mechanism in soil. So they are also present in the soil. Apart from that, some actinomycetes are normally present in soil. Also few algae can be present in soil. Some parasites are present in soil. So this number of microbes can be vary with respect to which type of sample we have collected and what is the soil composition of that soil. So depending upon that, availability of nutrition also affect the this microbial flora. So these are nothing but the factors. Now we have to discuss about the factors which are affecting the rhizospheric microorganisms. Normally we talk about the it is nothing but a soil which is related to the plant roots, and it is nothing but a rhizospheric region, and there abundance number of microorganisms can be present. So here various factors we have to discuss. So firstly we have to discuss about the proximity of soil to the root. It is nothing but a closeness of that soil with root. Normally microorganisms can easily grow where nutrition is available. That's why they carry out their life cycle. So that proximity of that soil to root is necessary. The number of rhizospheric microorganisms is greater near the root, and their number continuously decreases with increase in distance from the root. In last lecture we already discussed some essential organic substances are released from the plant roots or root tips, including amino acids, we can say vitamins, enzymes, and they are easily digested or made available for the metabolism to carry out their nutritional requirement of that microorganism. That's why microorganisms are more present near the rhizospheric region. But as distance from plant root increases, the number of microorganisms can be decreases as there is a scarcity for the scarcity of the nutrition. It is because concentration of organic matter which are released by the plant roots decreases with increase in the distance from the plant root. As they are normally secreted near the plant root or plant tips, and they made available for the microorganisms which are related or with close to that plant roots. But as distance increases from plant roots, that nutrition which is available for the microorganisms might be decreases. That's why the proximity is major factor which affect the microbial population in rhizosphere. Another factor is nothing but a temperature and light intensity. It is again another factor. Sometimes for the microbial growth or for optimum growth, microorganisms require an optimum temperature so that they carry out their metabolic processes so easily. So here, low temperature and low light intensity decrease the rate of exudation secreted from the root so that number of rhizosporic microorganisms decreases. As low temperature, that means below optimum temperature you can say, and low light intensity. So also it affects the photosynthetic rate of plants. So due to that, the secretion of plant roots can be decreases from the plant root. So that due to that, the organic substances which are required for the microbial nutrition may be decreases. That's why it is again another factor to affect the microbial population near to the rhizosphere. Another, on the other hand, number of microbes in rhizosphere increases when temperature and light intensity increases as multiplication rate is high. Again another factor. So as temperature lower down or light intensity lower down, secretion from plant root decreases, so it is adversely affect on the microbial population. Apart from, if light intensity increases or temperature increases normally, it also affect the normal metabolism of microorganisms. Again there is a decrease in the microbial population. So normally it is nothing but for the microbial population it required optimum temperature so that microorganisms can easily survive in that environment. So it is again another factor. Now it is third factor, type of soil. Which type of soil it is? It also affects the microbial population at rhizosporic region. The number of microbes in microbes is high around root is in sandy soil. So here again we can discuss about the type of soil. Which type of soil make available nutrition for the microbial growth. For example, R is to S ratio. It is nothing but a number of microorganisms present in that soil. That ratio we can calculate by means of R is to S ratio. It is nothing but a number of population of microorganisms present in that rhizosporic region. We can calculate by means of that R is to S ratio. It is very high in sandy soil and low in clay soil, as it is because sandy soil contains very little organic matter and root region is only place where organic matter is available and microorganisms can grow. So with respect to type of soil, microorganism population in the rhizosporic region can be affected or changed. On the other hand, in fertile soil such as clay soil, organic matter is sufficiently available for the growth so that microorganisms need not to depend on the root exudes for the growth. As we normally say, light intensity or presence of light also affect the root secretion and it is adversely affect on the microbial population. But another factor, as again we differentiate clay soil and sandy soil. Clay soil is already rich in organic substances. That's why microorganisms extract their nutritional requirement from the soil, and they shouldn't depend on the exudes from the plant source. So they cannot carry out their normal metabolism by absorbing the nutrition from that clay soil. Another factor is nothing but the age of plant. It is again another reason microbial population can be affected. So normally you can say crop plant, it has a limited time period or lifespan, including Cajanus cajan, you can say soybean or green gram, black gram. So they have a lifespan of about 3 months or 4 months or 6 months, we can say. Due to their lifespan, at the time of their growth, their photosynthetic mechanism normally carried out and they release more organic substances from the plant root. But as their life cycle is completed and they are now starting to wind up their life cycle, so release of that nutrition from plant root is minimized and it again adversely affect the microbial growth in that rhizosporic region. So with respect to the type of plant, it again varies. Types of plant and location of root. It is again another factor, which type of plant is and what type of location is there. Root cap and region of root from where lateral root arise are primary site of exudation secretion. These are nothing but a hair-like appendages invade from the plant roots and they are responsible for the release of some exudes from the plant roots and they acts as a source for nutrition for the microbes. Amount and type of exudate secretion differ with species of plant that influence growth of rhizosporic microbes. For example, some plant root release antimicrobial chemicals such as glycosides, hydrocyanic acid and several antifungal agents that inhibit rhizosporic microbes. So it is again another limiting factor for the microbial population at rhizosporic region. So normally you can say in dicotyledon plants, nitrogen fixing organisms have their association with their plant roots. So normally they absorb nutrition from the plant root. In other hand, they absorb or fix the atmospheric nitrogen and plant make available for the plant. Apart from that, some species of plant they have some secondary metabolites or we can say phytochemicals released from that plant root which acts as an antimicrobial or antifungal substances so that they inhibit the growth of microorganisms so that it is again acts as a limiting factor for the microbial population into that rhizosporic region. It is again another species dependent or plant specific factor depending upon which type of species which produces antimicrobial and fungal substances which affect the microbial population. Apart from that, some microbe and plant have mutual association with plant roots. Depth of root, it is again another factor. In general, number of rhizosporic microorganisms decrease with increase in depth of the root, which is mainly due to the anaerobic condition. So normally it is mainly due to the respiration. As we are moving down toward the earth crust, availability of oxygen for the respiration decreases. That's why it is quite difficult for the microorganisms to survive at particular depth. So again, it is normal parameter which affect the microsporic rhizosporic microbes which are present in that particular environment. Root respiration. Plant root release carbon dioxide during respiration that make the soil acidic. It is again another adverse affecting factor on the microbial population. So due to that acidity, yes, microbial population can be decreases into that particular region. So it is all about different factor on the microbial population. So what are the role of rhizosporic microbes in that particular rhizosporic region? We will discuss. Rhizosporic bacteria such as Rhizobium, Azotobacter, Clostridium, these are or they are involved in the fixing atmospheric nitrogen and make it available for the plant. So in other hand, they absorb the nutrition from the plant. So it is nothing but a symbiotic association between plant and plant roots and microbes. Phosphate soluble microbes such as Bacillus polynexa release free phosphate from the inorganic salt of phosphate, which is important nutrient for the plant root. Again, phosphate soluble bacteria which are also helpful for the plant root. Several rhizosporic microbes including Azotobacter, Ultrabacter, Pseudomonas, Agrobacterium, they produce growth hormones such as gibberellin, indole acetic acid, and which are involved for the proper plant growth. Or these are nothing but the growth promoting hormone produced by the microbes and they are absorbed by the plant. They are useful for the proper growth of plants. Many rhizosporic fungi including associated with the plant root in the form of mycorrhiza, whether it is endo or ecto. Mycorrhizal fungi promote plant growth by various ways. So these are nothing but association we will discuss, either it is positive association or negative association. We will discuss in this lecture further. So it is all about the role of microbes or rhizosporic microbes for the plant growth. Next topic we will discuss in next.